Dr. Juanita Anders has joined us here, and we're thankful that she can be here. She, as I mentioned, is really an internationally recognized pioneer in photobiomodulation, again, PBM. She is a professor of anatomy, physiology, and genetics at the Uniformed Services University. If you haven't heard of the Uniformed Services University, it is considered the West Point of medicine, and it provides both the rigor and a curriculum tailored to the military, so serving their needs. As a scientist at the nation's Federal Health Sciences University, Dr. Anders conducts research that is really vital to the survival and recovery of our U.S. military and VA populations. She spent her career really at the forefront of light-based medicine, moving it from the fringes into the core of military and veteran recovery programs. Her research has proven that specific wavelengths of light can trigger cellular repair, reduce inflammation, and effectively treat chronic pain. She's here today to help us understand how we can use light to jumpstart the body's internal healing mechanism and how this technology is currently serving our service members and the public alike. Welcome, Dr. Anders. Thank you.
Thank you for having me. I'm really excited about this opportunity to talk about photobiomodulation.
So your research at USU has essentially turned light into a biological drug. So I would love to turn the floor over to you, let you walk us through your slides and explain the basics of PBM and specifically how photons interact with our neurons to kickstart the healing process.
My pleasure. All right. So as I said, I'm excited to be here and share our experiences. In these few slides, I'm really concentrating on the basic science and background because I think so many people have heard of red light therapy or light therapy, but they have a lot of basic questions. So I'd like to spend time on that. So I don't have a comprehensive look at the research we've been doing to modulate pain, but I'm glad to discuss that after these few slides. Sorry, there seems to be a lag when there we go. So these are the disclaimers. I really want to just draw your attention to the last one. As a federal government scientist, any opinions I give today are mine and not those of my university or the Department of War. So I want to tell you about photobiomodulation therapy, but first I'm going to tell you what it isn't. And that is any light that affects your body through your visual pathway. We're not using the visual pathway to cause changes in your body, although that's a very, very powerful, as you realize, method, such as with circadian rhythms, how people suffer from SAD during the winter months and do a light, bright, usually white light therapy to overcome that. I'd like to introduce a few terms to you. First of all, let's look at optical radiation. And optical radiation is part of the electromagnetic spectrum, and it consists mostly of UV, visible, that's the light we can see, and the infrared. When we talk about photobiomodulation, we're talking about generally, for deeper bodies, using light in the visible red and in the infrared A, which is also called near infrared. And so the common wavelengths are from the 600s to about 1,400 nanometers. So if we look at the definition of photobiomodulation, that is using light, and that light is non-ionizing optical radiation, which means it doesn't have enough, those wavelengths of light we use do not have enough energy to remove an electron or from an atom or molecule. And they do not damage DNA as some of the UV wavelengths can. As I said, we're going to be looking at the visible and near infrared spectral ranges. And this light will be absorbed by endogenous chromophores. Now, chromophores are molecules that can absorb specific wavelengths of light and then cause a biological, photophysical, photochemical event within the body. And we're looking here at endogenous chromophores. That means the chromophores that your body actually makes versus exogenous chromophores that are used like in photodynamic therapy, say to treat cancer, where you put in a molecule that is sensitive to a wavelength. That molecule then will tend to go into, say, a tumor zone, and then you use a specific wavelength of light to destroy the tumor cells. Now, if you've looked at all into photobiomodulation therapy, and it is a problem in the field with a lot of sort of fly-by-night devices, and they often try to distinguish themselves. They'll say, this is LED light, or this is laser light, or, something to make themselves stand out. What I want to emphasize, although there are differences between lasers and LEDs, such as lasers have one wavelength of light, LEDs usually have a range, maybe 10 to 20 nanometers range. But once that light interacts with the surface of the body or tissues within, it's scattered, it's absorbed, and it acts the same. So don't be convinced by anyone that their specific device is better. It's the light that's doing the treatment, not the device. So if you want to get light into the body and treat, say, spinal cord, nerves, brain, you want to pick a wavelength of light that isn't fully absorbed by major substances in the body, such as blood in the red line here, melanin, which is degree of pigmentation in your skin, and water. And there is a nice range in, especially, I'll show you in a minute, around the 800s that is very effective in getting to depth of the body. So we published this back in 2005. It's amazing how quickly time flies, but my laboratory was the first to actually measure light entrance into anesthetized, in this case, rodents. And we do this as a standard in my lab. Usually before that, light was mathematically modeled. It's effective. It's still used today. Or they had what are called phantoms, where they would make comparable sort of tissue-like material and look at light. But in this case, we had the living animal anesthetized. We put bright white light of like 500 to 1,200 nanometers on the animal, and then using a fiber optic inserted through a hypodermic needle, we were able to measure what wavelengths of light got into the body from the skin just below the dermis down into the spinal cord itself, because we were preparing to use light on spinal cord injury. What we found at that time, and it's kind of defined what a lot of devices use, is that the wavelengths that go the deepest range from about 760 to 820 nanometers, with the peak at 810. And the beauty about 810 is that it is at what's called the isobestic point of blood. That's oxygenated and deoxygenated blood. And it has high transmission through blood. It also has 98% transmission, as we've measured, through water. So it's great. Water and blood in your body are not going to absorb it. And it's also highly absorbed, if you see in this transmittance graph, by the brain and spinal cord and muscle. And these tissues are very high in mitochondria, and you'll understand that statement in a minute. I'd just like to shout out and say that my colleagues that worked on this with me were from the FDA, the Institute for Device Development, and they're both wonderful physicists and electrical engineers. So there are two things I want you to understand about light for treatment. There are two important factors. One is wavelength. As I just said, you want to choose a wavelength if you want to get to depth that is minimally absorbed by water, blood, such as 810. Another important thing about wavelength is the wavelength determines the depth of penetration of light. It's exclusively the wavelength. Now, if you look at this panel of pictures, you see a hand and light being transmitted through the hand, and then the other panels are infrared cameras. And the number of photons that you deliver along the path that's defined by the wavelength, that's the depth, is determined by the output power. And so we'll talk about things like irradiance, which is milliwatts of light or watts per centimeter squared, and fluence, which we really talk about as the dose. And that's joules per centimeter squared. So that's essentially equal to power density times time. That's how long your treatment would last. I'm sorry, a message had come up and I was reading that. So optimal laser parameters vary with the location of the target tissue. Now, this sounds like common sense, okay? An important advance that was made in this treatment area was realizing that you needed to optimize the amount of light you deliver at the structure you want to affect. And that sounds like common sense, as I said, but for a long time in this field, because this field started with treating wounds, burns, surface, or studies in tissue culture dish, that when they, in the late 80s, 90s, wanted to start to treat, say, pain, they were, one study targeting the stellate ganglion. They used the parameters that were successful in the tissue culture dish or on the surface. And so there was a whole slew of negative reports that were published in that time frame because people didn't realize the amount of absorption and scatter and some of these principles that I'm talking to you about now. One thing I want you to keep in mind if you're considering a red light therapy device is it doesn't have great depth of penetration. And also, you lose about 30% of that power just by that light going through the skin. So if you get a very low level device, it's going to be even lower as it passes into kind of a shallow depth. So you're not going to want to use that for affecting deeper nerves, brain, spinal cord. So the first law of photobiomodulation, photobiology, which is very key to this field, is that for light to have an effect on your body, the photons must be absorbed by a molecular chromophore. And I already mentioned to you that in PBM, we utilize the endogenous chromophores to absorb light of specific wavelengths. Now, in the first panel on the left, you see a neuron, and then inside of there are mitochondria. And the middle panel is the inner membrane of the mitochondria, showing the electron transport chain. And the primary chromophore that people are exciting, there's a lot of evidence for this. It's not 1000% solid, but people are looking at some other mechanisms for especially different wavelengths of light. But the main one is cytochrome C oxidase, and it's the primary photoacceptor in the wavelengths that I'm talking about. So it the cytochrome in the mitochondria roughly will absorb between 600 to 1,000 nanometers. And it's actually absorbed by copper centers within that molecule. And the little graph on the side, just so you realize, there are sort of two peak zones, depending if the coppers are oxidized or reduced form. And these peaks occur in this red light, 620 to 680 wavelengths, and in the 760 to 850 nanometer wavelengths. Now, what happens when the light is absorbed? When it's absorbed by those chromophores, it upregulates the activity of the mitochondria. It's a lot of times in injury and disease, there's a downregulation in the functioning of the mitochondria. And this upregulation increases ATP and it increases reactive oxygen species. Not too much. If you put in too much light, you'll get too much, and that can be destructive. A lot of people also talk about the generation of NO. There's a lot of controversy about the effectiveness of that. NO is very short-lived. It has a very short half-life, and it reacts in the local environment, so it doesn't have a broad effect. So the main pathways are changing things like the ATP affecting cyclic AMP and the AP1 pathways to the nucleus, whereas the reactive oxygen species, or ROS, will stimulate the NF-kappa B pathways. And what does that do? That affects gene transcription. So you ultimately get changes in RNA and protein expression, and these can initiate cellular signaling, which could lead to improved clinical outcomes. Now, early on, this field was called biostimulation because everyone talked about that this was a stimulatory effect. We now know that when you put light on, you could get an upregulation of some genes, a downregulation of some genes, and no change in other genes, say like structural genes. But let's say that your nerves are in a very inflamed state. When you put light on them, you will get a down regulation of the upregulated inflammatory markers, and you will get an upregulation in anti-inflammatory markers. So what you get depends on the state of the cell. Here's an example of a result of some of the work we did. I told you we used light on acute spinal cord injury in rodent models. And what we found was if we used it immediately after injury, that we altered the migration patterns of cells into the injury zone. So we got a decrease in like the chemical that attracts macrophages. And then the macrophage population fell. So they didn't make factors that would attract fibroblasts or other glial cells and lead to the scar. So by breaking that pattern, we blocked the secondary cascade of injury that you can get in spinal cord and brain. And this led to a decrease in scar formation and an increase in regeneration across the injury zone in this animal model. When we looked at the gene changes, we looked at like 1,000 genes, and we found that with the light and injury that we were able to alter 200 genes significantly. And some of these were upregulated, as I told you, and some were downregulated, and some weren't changed. But we did see a lot of changes in decreasing inflammation, decreasing glutamate receptors. And I'll just walk you through this little study. We were looking at normal human neuronal progenitor cells in culture. And what we always did in our lab was kind of do this little plated out irradiance, that's milliwatts per centimeter squared. And then we did different times, and that gave us a different dose here in joules. And what we found when we did this, we measured neurite extensions, and we found there wasn't just one combination of irradiance and time to give us an effective dose, but there were groups or clusters that you could choose from. And we went on to do this experiment. Now, normally when you grow normal human neural progenitor cells, you have to put them in what's called a factor media. So you put in a lot of different growth factors to get them to differentiate. So we did a study where we had the cells just in the media without any additional factors. We had the factor media, and we had controls with nothing, and then light treated. And what we found was that the light treated and the factor treated gave a comparable result in neurite extension, and both were better than control, of course. So when we looked at the genetics for this, we found that what the light did was stimulate these neural progenitor cells to produce their own factors that they needed to begin differentiation. So we had upregulation, fibroblastic growth factor, and especially nice increases in nerve growth factor. So this is my last slide. I see some of you thought this was too technical. I'm sorry about that. I guess that the main points were you really have to think about wavelength and the amount of light that a device gives and what you want to treat. If you want to treat surface wounds, pretty much red light will be effective at lower doses. Now, I've done a lot of work on pain pathways, and I put those here. There are two major ascending pathways. One is called the dorsal column pathway, and the major neurons that run in that pathway are heavily myelinated neurons. They're called A's and B's. And they transmit conscious proprioception, so you're aware where your body is in space, two-point tactile discrimination. While the other pathway it's called the anterolateral spinothalamic pathway, and that conveys your information about pain and temperature. And what we found in our research, and I'm glad to discuss any details during the question and answer, was if we used lower irradiances, we could modulate pain. And oftentimes we measured the dorsal column pathway using von Frey. And we were able to decrease pain responses. And the major mechanism really was a decrease in inflammation. And the interesting thing about pain, as I'm sure all of you are aware, when you get inflammation and you have injury or even dieback by the skin, you will get an inflammatory response along the entire ascending pathway up to the brain. So if you can manage to decrease the core inflammation, that will start to downregulate the inflammation along the ascending pathways. Now, the other thing we found is if we used very high irradiances, and I'm using watts per centimeter squared delivered at the nerve, we can block pain transmission. We just stop the pain transmission. And these are affecting, this treatment affects specifically the lightly myelinated and unmyelinated pain fibers. It has no effect on the large myelinated somatosensory fibers or on the motor fibers. So we've done another test, and my colleagues at Case Western have done tests looking at motor function. And so it's a very nice way to take out pain fibers, and this is transient, it's not permanent, which is great because you want to break that cycle of pain transmission, decrease inflammation, to get a result or a change along this ascending pathway. And often we recommend doing the block if it's severe pain, followed by the lower parameter of lower radiances to cause other changes like decrease in inflammation. So that's my last slide, and I'm glad to go on and enter the discussion phase, Sherry.
Good. Thank you, Dr. Anders. We have a range of individuals here participating. So you've got people who are in the profession of physical therapy, occupational therapy, speech therapy. So, and then you've got individuals who, are working through their own neurological challenges. There are quite a few comments that they love the amount of detail. So we appreciate what you were sharing. And I do think that it's very valuable, even if you're not familiar with all of those terms up front and you're just learning about photobiomodulation. It's important to have that, and you can go back and view the recording for this, because when you start to go out and evaluate providers who are doing this, those are the kinds of questions you're going to want to know, and that arms you with the information that you can have in your back pocket to make sure you're getting the right kind of service for what you need. I will say, if you take this to a general practitioner or a neurologist, they're not going to know in most cases, this information. And so again, this is the kind of information you'll want to share with your care team if this is something you want to consider. So I do think that information is extremely valuable. So thank you for walking us through that. Now let's take it and break it down into some actionable items for our audience. Let's start here. I think this is probably what's on everybody's mind, right? You've got all of these devices that you can buy off of Amazon. You can buy from all of these other producers. You've got influencers out there using them, recommending even for your face, for anti-aging, for pain, for athletic performance. So can you just walk us through, for pain, if you want to buy a device for home, is it worth it? What you should be looking for, and how does that compare to what you can get from a provider with the higher performing devices?
I think for things like chronic pain, back pain, you really need to go to the higher powered devices, and you're not going to get those as a home unit. The FDA has strict limitations on the wavelengths and the amount of power you can have at home in your device, and they're safety driven, so you don't damage your eyes or burn yourself. So for example, we did a study with my two colleagues down in Sao Paulo, Brazil, looking at back pain. Now, normally they were doing a treatment using lidocaine injections for back pain at the L2 lumbar 2 level of the spinal cord. And they got funding to do this small pilot where they did the lidocaine injection, they did radio frequency, and then they did light, and it was 808, very close to 810 in that high peak, delivered though through an optical fiber, a very small fiber, and it was put in through the same needle they would use for injection. And what they found was, with a few minutes of this treatment, that the patients had comparable results with the light and lidocaine. And for some patients, the result lasted for 21 days, and you didn't have any other drug side effects associated with it. Both of those therapies were superior to the radio frequency that they tested in Sao Paulo. So for most cases, remember I said to you that in the past, people would use a low power on the skin, and they think they were going to affect, say, arthritis pain. You just didn't get sufficient degrees of light on those chromophores to make that change, to change the genetics and block that pain. So when we started, we're funded by the military to work on a mobile device using these high output power devices delivering high power at a nerve. And this would be for in austere environments. And we're doing some pig experiments in Israel right now. And for those experiments, we found that the amount of light we would have to put on the surface, and this is the same for people, it would be very high wattage and it would cause burns. So most of deep, like spinal cord back pain, targeting dorsal root ganglion, you would want to do through a fiber inserted through a needle, and you would need a high power device. The home devices, if I may add, I said most of them are suboptimal. You'll get maybe a little effect, but not a good effect, especially with pain. But for other things, I think doing upregulation of collagen in skin, red light works. There's a new face device that came out recently that has the best power or dose to do that. Those are useful, but for hardcore pain control, unless maybe you have just the loss of nerve endings going into the skin, like with neuropathic pain that you get
Mm-hmm.
sometimes, that in that case, you have a chance because that's right under the dermis to get various wavelengths of light in and cause those nerve endings to regenerate. But in that case, it's not going to change the inflammation and you're just going to get persistent dieback. Like you treat.
Okay. Okay, so if somebody wants to investigate this for pain, where should they go, Dr. Anders? Because there are clinics that in almost every single city. I am in Atlanta. I looked it up. There's probably 7 or 8 in my 20-mile periphery that I could go to that advertise for pain reduction. Is that something we should explore? If so, what kind of pain is well suited for that? And for this chronic pain, for these issues, where should we go for that type of a treatment?
Yeah, it's a very good question, and it's hard to answer because the field has not defined all of these parameters. I think for things like back pain, there are some doctors who would do that and possibly use light. But at this point, light is not widely adopted by them. And part of it is that it's not covered by insurance. And that's why these topical treatments where you go in and get lower power repeated treatments where they'll give you a deal like 6, you pay for 4 and you get 6 on some things. Um, some, I think in the US, maybe chiropractors and physical therapists use it the most, but again, it's not widely accepted. So when it comes to light, I think it has great potential. I think it still has to develop and be embraced and accepted in the community to get that kind of guidance. And it's just not there now, I'm sorry to say.
Okay, so for somebody here listening then who wants to try it out, are you saying that a chiropractor or a physical therapist would not have access to the high intensity wavelengths that would enable it to be used for chronic back pain?
No, some of them absolutely do. And
Okay.
I'll tell you, there was a company that I did. I'm not just suggesting that the company sold their devices, but they were the first company to develop high power devices, and it was used in the vet clinic. And it's huge. Light therapy has been totally adopted for arthritis pain, all kinds of pain in animals, and widely used. Well, the company was called Light Cure, And they had started in the vet world, and then they expanded into the clinical human side. And they sold that company. But in the time that they were doing that, they had over 4,000 devices in the field and probably still do. And they collected patient data, and it was the best data set. And their devices went up to 25 watts, but they were based, I think, like I'm in Maryland, and there's a clinic in Northern Virginia that uses the high and low power devices. So there are some out there specifically for pain. And when people have contacted me in the past they'd say I'm in Atlanta or whatever. Is there someone who is using this that I could try? I would go to the company, and I still have a contact with the company they send it to, and say who in the area is using this high powered device, because I did a lot of prototype testing and I did trust that device, because it's really hard. A lot of devices don't produce the wavelengths or the output powers they say they have.
Okay, can you just review then the wavelengths that they need for the chronic treatment? What should they be looking for?
I would look for something in that near infrared in, say, 780. Some people go up to 820. That's fine. A
Okay.
lot of devices are around 810. Some people have shifted like to maybe 860 and the 1040s, but that's really for transcranial brain treatments. A lot of
Mm-hmm.
1040's been done. Um, so those are the wavelengths I would look for. I would look for a practitioner who has a high-powered laser, who can deliver at least 10 watts to deeper structures, um, that for that kind of pain treatment. Now, if you're looking at, and I know they sell it, a bunch of devices, they say for your well-being. I'm sure you or people who are on this call may have seen like light panels. And there's
Mm-hmm.
also these beds that look like tanning beds, but it's
Mm-hmm.
photobiomodulation beds. And they make a lot of claims, but they really haven't done the research to see if that's truly efficacious. Because they'll say, it'll drop your whole body inflammation, decrease pain. And so right now, I'm involved in a study at the Air Force Research Laboratory. And we just completed, I think it was 44 patients, looking at two different beds, one with a lower irradiance and one with a higher radiance, a lower and a bit higher dose. None of it's real high. But, where I've done so much work on light delivered, we really don't know. What if you put light all over your body for 20 minutes? Will that have an effect? And we're just starting the analysis of blood serum, saliva, a bunch of questionnaires to see if these beds are actually doing anything, because they have not investigated. And part of that study, we also looked at some devices that people will wear. A lot of that is for musculoskeletal recovery. So wearable devices. And we also looked at a very popular two panels. And they advertise on those panels that you could wear clothes, you could wear your gym clothes, and you could exercise, and you just stand in front of that panel. But when we actually measured what the panel was producing, they do say recommended distances, which are correct. So if you're moving around and you're out of, say, further out than 10 centimeters, you don't get the dose and you don't get the benefit. And if you have clothes on, it really decreases the benefit. And the other thing we found in panels was it only works right in the center, and if you're up close. So I love this field. I've worked for a long time to try and define it and have it accepted. But there are, unfortunately, it seems to be device driven without the science in most cases. And I think that's a good point for the people who are here today. If you're thinking about a device for pain or anything else, you really want to find out, I guess, three things: the wavelength, and is it really, you can't always trust that wavelength, the output power that you would use. And, did that company actually ever do an experiment with their device on a patient? Did they ever really look to see if it was effective? And most of them haven't. They'll say, oh, this device is good for changing inflammation, for increasing proliferation, for increasing circulation. But they just took what's in the literature. They never really tested their device to see if it was efficacious. There is a
Okay.
little, I just want to say
Sorry.
that there's a study done by a Dr. Mark Cronshaw. I met him 2 years ago in London, and he was working on he's a dentist, but he's working on his PhD. And he bought, I think it was 4 devices off of Amazon,
Mhm.
and he's very good at measuring light. And he tested those devices on what they were actually outputting. And none of them were what they said it was, unfortunately.
Okay, well, I think that is fantastic information. Well, maybe not what I was hoping to find out today, quite frankly.
I know.
So I think most of us, because I was under the impression coming into this that they were very widely available and being very effectively used. And what you're telling us is that for severe chronic pain, it is available, but you've got to really do your research on the device that's being used. And do they have the research to do it? And have they tested that individual panel, not just general research on wavelength? So it's panel specific, it's wavelength and output specific.
Yes.
Okay. All right. Well, thank you so much. I want to encourage everyone, please, you'll get a poll here in just a second to rate our conversation today. I think we've got time for one more question. I've gotten several here pieces of information about concussion and skull fractures with light therapy. How would you summarize, Dr. Anders, the ability to treat that? So headaches and pain with skull fractures in that area.
That it's well known, and there are a lot of devices. Again, they run that gamut from some that aren't tested, but there are some very good devices uh, for that you could use for all kinds of treatment of the brain. I'd like to tell you there's a Dr. Paulo Casano at Harvard, and he runs most Fridays, he'll call it a sort of a brain clinic where they talk all about different devices and uses of PBM. I know firsthand that on the military base, we were they were using one of the devices that I had worked on with Lightcure at the time on our wounded warriors. They didn't want to put light directly on the head, but they were treating the back of the neck, the temple area, and they got a wonderful relief from headache, chronic headache. So there is a lot of work done, and I think it's quite effective on headache. That's been proven that it increases circulation in the brain. And also there was one study out of Harvard where they put light on the brain and it decreased pain in the pathway. So it wasn't repeated, but that's an interesting study with this whole system connected, that might be a feasibility in the future.
Yeah, so it sounds like it's a promising future. There is benefit from photobiomodulation, but you've just got to be very careful in who you go to and what for and validate the claims that they're making. Is that
Yes.
fair? Okay.
That's absolutely fair, yes. And I
Okay.
wish I could just say go see this doctor and you'll have a wonderful treatment and device. But the field's not quite there yet.
Yeah, it's just not quite there yet. So yeah, use caution. Like I said, I've got literally within 20 miles of me, there's 7 or 8 locations that I could go to. I've done the research on several of them and have recommended several people locally to go and use those. And I have one at home that I use for different things. And I have found it beneficial, quite frankly, from that. But it is in the wavelengths that you talked about, Dr. Anders. So I was glad to see that I bought something that was, I spent a lot of money on it, and now I'm glad I did from talking with you. Okay, well, thank you very much. We'll be talking to you soon, and thank you for the research that you shared and what you're doing to
And I
move the
saw
field
some
forward.
questions such as the name I just cited, if there's any headache, or I can, gladly provide you with them
Okay,
after this.
great. So yeah, so I'm sorry to interrupt you there. Your contact information, then we'll make sure we put it in your bio on our website. You can reach out specifically. Thank to Dr. Anders. Thank you for offering that, Dr. Anders. And she can
Sure.
answer your questions directly. I appreciate you taking that responsibility on. Thank you.
You're welcome.
Okay, and you will get a list of these questions that are on here. There are quite a lot of questions, and so you will get those questions, Dr. Anders. So when people reach out to you, you'll have that. Okay, thank you so
Okay,
much. We'll
excellent.
we'll we'll be connecting soon.
Thank you for this opportunity. Bye.
Welcome. Bye-bye.